Hi everybody, welcome to another Chats with Chaudhry. I hope you're safe and well. Today I'm continuing a series I'm doing with RSSL looking at the different areas of analytical testing. And today we're going to be looking at particle characterization in pharma with Caroline Welch, who is the Senior Scientist 1, and Wendy Hulse, who's Senior Scientist 2 in the Physical Sciences Department at RSSL. So how are you both?
I'm good, thanks.
I'm good. Yeah,
Excellent.
also good, thank you.
I'm glad to hear it. And so, where are you guys calling in from? So, Caroline, where are you calling in from? So you look like you're not in the office today.
I'm not. I'm calling from my dining room.
Excellent. Wendy, where are you calling in from?
I'm on site at the Reading site.
Fantastic. All right. So, before we start talking about particle characterization, could one of you give me a quick overview of RSSL and then also give a little background on yourselves as well, if you don't mind? So, Caroline, would you like to do that?
Yeah, that's not a problem. So you gave our really RSSL specific job titles, Senior Scientist 1 and 2, and that's within our Physical Sciences Department at RSSL. But RSSL overall is a contract research organisation for both the food industry and the pharmaceutical industry, helping anyone to develop new products, solve problems with their existing products, just routine testing. So we're there to help anyone who needs a bit of science done.
Fantastic.
Myself, I did my chemistry degree and master's in London a few years ago, and I actually came to RSSL straight afterwards. So I've now been in the Physical Sciences Department looking after particle characterization for 6 years now, which is a crazy amount of time.
Excellent. And Wendy, your background?
I did a degree in chemistry and then I did a PhD in pharmaceutics and drug delivery. I've worked in both academia and industrial settings, so quite a broad range. I've been at RSSL nearly 2 years now.
Fantastic. So let's start talking about particle characterization. So Wendy, this is to you. First question is, what is particle characterization?
It's quite a broad range, really. It's mainly looking at size and shape of particles, but it can also include things like structural analysis. But today we're just going to focus on particle sizing.
Right. Okay. Well, then my first question now is that what is particle size, or why is particle size important for pharmaceutical formulations?
So I'll take that one. I guess the most basic answer is because all the regulatory bodies say that you have to. They say that you have to test it. They say that you have to care. So everyone does particle size testing. But the reason that is because, well, we take pharmaceuticals all the time and everyone thinks, you know, from the picture that comes into your head is a tablet and that's your pharmaceutical. But actually, it's everything. It could be a cream or a gel or your inhaler, your eye drops, your eardrops, kind of, they're all actually particulate systems.
Sure.
And the way that being a particulate system has an impact, the most important one is the bioavailability. And the particle size of your drug within that product will affect how quickly it gets into your body, how much of it gets into your body, and all of that obviously impacts whether it's going to have the effect on your body that it needs to and make you better. But there's also a whole list of things that the particle properties affect, like the stability of the product, whether the product is uniform, the appearance of the product, whether you can even process the product and make it. So many reasons.
Right. Wendy, have you got anything to add to that?
Yeah, so I'll just sort of give an example. If we look at the tablets that Kaz mentioned, so things like mixing, granulation, compressing the tablets, getting the coating on there is affected by the particle size. Also affects the tabletability of a product. If it's not uniform granules, it might not tablet properly. And the dissolution rate is also proportional to the size of the APIs.
Right. Okay. So how do you go about measuring the particle size distribution then within a drug?
So let's start on this one. There's a variety of methods, but laser diffraction is ubiquitous and it's referenced in pharmacopeias.
Right.
It's not without its flaws. It's a volume-based technique, so it's very sensitive to coarse particles, so it can be skewed. The data can be skewed. If you've got one large particle, it can affect your results. And it also assumes that all particles are spherical. So if you've got things like needles, you might get two different distributions. Here at RSSL, we've got master sizes, so we've got 2000 in the newer model, which is a 3000. I think Kaz wants to.
No, so I was just going to say, like, we use the Malvern master sizes because they are everywhere. Malvern Panelytical, who make them, are very effective at distributing their instruments all over the world. So we kind of keep up with that and use that laser diffraction technique, because it works for so many industries and so many clients. And as a contract research organisation, we want to help as many people as possible. But we also do loads of microscopy and lots of image analysis on that microscopy. There's really basic techniques like sieving as well, which can also be really helpful in sizing particles. But one of the things that Wendy mentioned is the master size of 2000 and the master size of 3000, which are basically old and new versions of the same instrument. What we spend a lot of time doing now, especially this year, is transferring from that old instrument to the new instrument, just to keep all our clients really up to date on their methods. Because after early next year, the master size of 2000, kind of, there isn't going to be support for that anymore. All the spare parts are running out. If anyone is reliant on that, then they're going to be a bit screwed when it comes to their analysis. So we are, I know, it's a difficult situation. So we're doing loads of work to take all those methods from one instrument, make them work on the newer instrument, which is sometimes incredibly easy because obviously they work on exactly the same principle. Sometimes it's not, because the geometry of the new instrument is a bit different. All of the algorithms inside have changed. In a lot of ways, it's much better. But we often are re-optimizing these methods to still give the same results, even though it's a different, newer piece of kit. And then people can be happy, have peace of mind that their testing can continue on for years to come.
Okay. Something comes into my mind though. You know, hate me chucking a question in that you're not expecting, but when you are measuring particle size distribution, what is the biggest challenge though you face when you're doing the measurement process? I mean, you said you've got different methods, but what is the sort of most common challenge you have when you're trying to get those measurements right?
Hey, we might give different answers. Maybe we should both answer this one.
Yeah, well, from your point of view, Karen, what would be the biggest challenge for Karen? Would be the biggest challenge for you?
I would say for me, it's understanding what aspect of the particle size is the important one. So like Wendy mentioned, the Malvern master sizes and laser diffraction as a technique as a whole assumes that everything is a sphere, which is really nice and easy, because then we just have the diameter of a sphere, one number, it's really nice. But then for some applications like making tablets, making substances that need to flow, like thick pastes or anything like that, the shape of the particle is really important. So it's actually kind of discussing with the client and saying, are you okay if we use a technique where everything is assumed to be a sphere? Or actually, do you want us to use a technique because the longest length is more important? Or maybe the ratio between the shortest width and the longest length, maybe that is the most important size parameter. So there's so many different sizes that you can use. It's not just size. And so for me, the hardest bit is understanding and deciding which one to use. But you always have that conversation to work it out.
So just following on from that though, because you said the sizes, it depends what you're measuring, and you said you talked to the client. So is that more dependent on what the client needs, or is that what the Pharmacopeia has said you need to measure for that particular sort of API or drug?
Again, it really depends. Like, often laser diffraction is specified for that material, and that assumes it's a sphere, so we absolutely have to. But for the pharmaceutical industry as a whole, particle size analysis isn't necessary, because knowing that your particles are 5 microns in size is really, really useful. It's understanding how the size of your particles relates to how fast they dissolve, or how those particles flow when you're trying to make it. So it's not like you want to know the size just for the purpose of knowing the size. It's all about correlating it to something else, like how you process it, the consumer experience. So yeah, sometimes we just have to do laser diffraction. But then a lot of the time it's understanding about what the particle size is going to mean for the product.
Fine. Okay.
And therefore what type of size we have to measure.
Sure.
Yeah.
And Wendy, what about you? I mean, is that the same challenges for you that Caroline mentioned, or do you have, is there something else which comes into your mind which you think is a difficulty that you have to overcome when you're doing these sort of measurements?
I think I was going to say something similar to what Caroline's already said. And I think it's not just the size, it's the shape as well, because that can affect the measurements.
Fine. Okay. So also, I mean, do you just measure the particle size of drugs and APIs, or are there other things that you measure as well?
Well, we measure the excipients as well. So if you've got a pharmaceutical, I mean, you might have the most amazing API in the world, but if you
Yep.
can't get it into the body by a delivery system, so a tablet or a liquid or a suspension, then it's absolutely useless as a drug.
Right.
So we had things like bulking agents, binders, disintegrants. A lot of those are picked specifically because of the particle size. And if it can't work properly, then we might have to modify it. So we could like mill it or grind it to get it to work, to get the right size. So imagine, say, you've got a box of muesli at home.
Yeah.
If you shake it up, you've got all the big chunks at the top. You've got the sediments at the bottom. You get similar effects in APIs when they've been manufactured.
Right.
So we do know that happens. And obviously, you either get a massive dose of API or nothing if it's not, if the particle size isn't controlled properly, which obviously you don't want. Sorry, go on.
No, I was going to say, no, it's a really great way of explaining it. So I'm visualizing, I had my Alpine this morning, actually. So I'll be visualizing it straight away. So that's excellent. Cool. Sorry, I stopped you. Is there anything else you wanted to add to that?
So yeah, it's not just particle size, it's things like the surface properties. So roughness as well can have an impact, how you design your system.
To say, like, it's like Wendy was saying with the muesli, kind of, you can either make everything exactly the same size so that when you shake the box of muesli, you don't get the separation out between the big and the small, or you can design kind of totally different things. You could design so that one of your excipient has a really kind of rough pitted surface, and then your tiny API particles fit into all the little pits and grooves on the surface, and then you get a nice blend that way instead of having all the particles exactly the same size. And I'm so glad I don't work in kind of pharmaceutical formulation because it's so complicated and there's so many things you can do. But they're the ones who kind of have to come up with their kind of designed and engineered ways of making kind of a uniform blend and making everything work with their particles. And then we just come up with ways of checking that it's working and helping to solve the problem.
Fantastic. Well, no, thank you for that explanation, both of you. I mean, that's been really interesting to hear more about that. So what are the current trends then within particle measurement sense? I mean, what are the trends that you're seeing at the moment, and how do you think things will develop going forward?
There's actually so many. I'm going to give a couple, if that's okay.
Sure.
So one of the first trends, which has actually been going on for a long time, is this move away from just size to looking at size, but the rest of the particle properties as well. So looking at the shape, looking at the surface roughness, the surface charge, the amount of water that it takes up, kind of all of those kind of things.
Right.
And not just being reliant on the size. So that's one of the trends. And like one of the newest fun things that we can do now has a very fancy name, and it's called morphologically directed Raman spectroscopy.
Right. Okay.
I know. And what all those big 4 words all just mean is that even in your drug product, so your API plus all the excipients like Wendy was talking about, you can disperse them, spread them all out nicely, and then look at them with this instrument. And it takes a really nice photo of every single particle. And so from that photo, you can determine the size, the shape, the aspect ratio, all of those parameters that we've been talking about. But it also takes a Raman spectrum of every particle. So you have your chemical information.
Right.
So you can get everything that we've been talking about for all of the different ingredients, your API, your excipient 1, excipient 2, separately, from analyzing them in the mixture all at the same time.
That's cool.
Which is a very fun piece of kit, and it's a very nice new toy that we get to play with. So that's one of our favorite current trends. But a much wider current trend is smaller particles. Particles have to be smaller and smaller and smaller. Kind of, I think
why
quite a long time.
Why do they have to be smaller?
Yeah, so many new chemical entities as drug products now. They either have really poor dissolution or really poor solubility, and so that bioavailability that I mentioned, getting them into your body, is actually really hard. And so people are kind of engineering their particles, and if you can get down to 100 nanometers, so really incredibly tiny, then you have so much higher surface area that things dissolve so much more quickly. If you can get down even smaller, then actually the intrinsic solubility can sometimes increase. So people are trying all kinds of things. Sometimes it's just get the particles, make them much smaller, so you have a nanoparticle formulation. Or you could make the particles really small and then hide them inside a liposome or a micelle, and all of these are different types of nanoformulation that mean the big particles are so much smaller, so that when you rub them on your skin, when you take them in a tablet, however, they get into your body so much more effectively.
Right, okay.
So yeah, make the particles smaller. That is a current trend.
Right. And Wendy, have you got any trends to add to that?
Sort of generic pharmaceuticals. So same drug substance is sold by different companies. So you've got the main supplier, and then you get companies come along and they'll try and make their own version. But they have to be proven to be biosimilar or bioequivalent.
Right.
So, and in some cases, it could be the particle size. So if you've got an inhaled drug, it needs to be similar particle size.
Right. Okay. And what are the sort of techniques then you'd use for those particular types of products?
Well, we've got our wonderful Malvern's systems. We've also got DLS, microscopic techniques. Very tongue twisted.
Right. Okay.
Yeah, I think all the same techniques that we have been speaking about, except instead of trying to correlate between the drug product and how fast it dissolves or anything else that's going on, we're trying to correlate between the new person's generic drug and the one that already exists. So it's like the same thing, but the whole focus is it the same? Because if it's the same, then it's fine. It can be approved for use straight away because the first one was approved for use. So we just have to prove it's the same.
Right. So without getting into brand names, maybe a well-known Coke brand and then a known label Coke brand, and comparing the two together, maybe, and see how similar they're.
Just like that.
Yeah, without getting into particular brand names. But anyway, all right. So thank you very much. I think that's all we've got time for, but that's really interesting. And I've learned a lot more. Hopefully the viewers have learned a lot more about particle measurements and particle characterization, as well as some of the techniques being used and some of the trends. So where can people get more information? I'd like to know more about what RSSL are doing and also the different techniques that you've been talking about today.
Well, I think just go to the RSSL website. There's lots of information, lots of specific information about the specific department that we work in. So head to that page. And you can also just email our inquiries at RSSL team as well if you have a really specific question, and then you'll get passed through to us.
Fantastic. All right. Well, look, Caroline, Wendy, thank you very much for your time. That was really interesting. I learned a lot in a very short space of time, so thank you for sharing. And I hope you as viewers found that useful as well. You can check out the RSSL website. It will be above the video, and also links to white papers. And if you've got any questions for Caroline and Wendy, because they've shown how knowledgeable they are already today, then feel free to leave any questions below the video for them. I'm sure they'll be happy to answer them. And I'm sure they're on LinkedIn as well, so you can send some messages as well. So Caroline, Wendy, thank you very much for your time. It's lovely to see you today. I wish you a wonderful week and a wonderful summer, and hopefully the sun will be shining where you are ready today, because it's a bit cloudy where I am in Surrey right now.
Yeah.
And thank you, viewers.
Thank you so much.
Okay, thank you.
Thank you. Thank you, viewers, for watching. I hope you found that interesting. Until next time, as always, stay well and stay safe. Bye-bye.